So first, we're going to talk about the spirochetes. Really interesting set of organisms. You guys are probably familiar with spirochetes because of the pathogens in this group, right? Treponema, Leptospira, Borrelia, these sort of organisms. It turns out these are important pathogens. But it turns out that most spirochetes are not pathogens. Spirochetes are common in a couple of interesting kind of environments. They're common in organic, rich, anaerobic environments. I'm thinking, for example, at the bottom of your Winograssi columns, there where it's black, where you've got polysaccharides being decomposed with some kind of anaerobic respiration going on. A subset of this environment are in the guts of wood-eating insects. And so if you want to see spirochetes, the easiest way to do it is to find an old stump, pull out a couple of termites, take the termite apart, and look inside the gut. And they're full of spirochetes. I'll show you a picture of that in just a moment. As common as spirochetes really are, there aren't that many of them in terms of their taxonomy. Part of this is an artifact. I mean, certainly the pathogens are well represented because people spend a fair bit of work on these. The free-living organisms are not very well studied. So unlike the proteobacteria that we talked about and the gram-positive bacteria that we did not, there really is a fair bit of homogeneity in this group. They're all heterotrophs. Most of them are microaerophilic or anaerobic. Most of them are saccharolytic, that is, they eat sugar for a living. A few of them will eat amino acids or fatty acids, but most of them are saccharolytic. Generally, hydrogen or CO2 are the product of fermentation. In other words, they dump electrons on hydrogen, on protons to make hydrogen, and some of them can make acetates. The morphology of these organisms is also relatively homogeneous. And so this, you know what spirochetes are shaped like, and that's common throughout all of this group. The organisms are generally long, rod-shaped organisms, which are twisted in one of two ways around this thing called the axial fiber. The axial fiber are flagella. These are polar flagella in which the flagellum never emerges from the outer membrane. So these things are gram-negative. Instead, it's trapped inside the periplasm. And the polar flagella from each end come together and overlap. Some of the small spirochetes have only a single flagellum at each end, so the axial fiber is 1 or 2 flagella wide, depending on where in the cell it is. Larger spirochetes have multiple flagella, like this one, which seems to have 3 or 4 of them. Again, down here, and then that forms the axial fiber. Rotation of those flagella cause rotation of this helical cell body, which moves through the medium. That's actually a lot more complicated than you might think. You could imagine, for example, well, we'll talk about motility at the end. No, we won't. We'll do that now because we're talking about it. So, I mean, if you think of this thing as a machine, right? If you've got a machine, a helical machine with a fiber that runs down the middle, rotation of that fiber gets the spiral nowhere, right? It just rotates inside the membrane. Nothing's going to happen. So how does it cause motility? How does rotation of this fiber cause motility? The way it does that is that, unlike most bacteria, the outer membrane is not attached to the inner membrane. Most bacteria, there are things called bare junctions between the two that fix the inner and outer membrane together. That doesn't happen here. And so when this thing rotates, the outer and inner membrane roll relative to each other. Now, in a frictionless environment, that would just cause the outer membrane to roll over the surface of the cell and nothing would happen. But it isn't a frictionless environment, right? And particularly at the scale these things live at, the viscosity of that water is really very high. And because the outer membrane is in a sense, glued to its aqueous environment, rolling of that outer membrane causes the inner cell to rotate. And as a result, you get this thing spinning, and that pushes it through the liquid. That's only true for those spirochetes that are actually helical. Some spirochetes are helical. Many of them, though, are a flattened helix. They're a sine wave. A little bit like a snake, right, laying on the ground. And so rather than a corkscrew, they're a flat sine wave. And these organisms, when the same thing happens, then the whole curve moves like this, just a little bit like the way a snake moves across a surface. And then there's some variations on this that we'll get to later. We already talked about habitat. Here's a great example. So here's a termite. You take a pair of forceps, not to be too gruesome, and hold its head and pull its body apart, and you get the gut here. This is the hindgut. Termites eat wood, right? And so, but animals don't have cellulose. So they can't digest this any more than cows can or anything else. They rely on the microbial community here to hydrolyze that cellulose, and the pectins for that matter, and all the other things. And then the insect can live off the products of that. It's actually not sugar from cellulose that insects absorb. It's acetate from the fermentation of the microbes that insects live on. This is true for wood-eating insects generally, not just termites, carpenter ants, carpenter beetles, etc. And so, if you take a sample of this hindgut contents, this is the kind of stuff you get, and you see there's spirochetes everywhere. This is a protist. I don't remember what the name of that thing is. You notice it's covered in these big, long flagella. It turns out a pretty significant fraction of these aren't flagella at all, but are also spirochetes. They have a particular docking system where they dock on the surface of the protist and in many ways function as accessory flagella on the protist. So it's a really interesting symbiosis. This led to the notion that flagella and cilia in eukaryotes may have originated from spirochetes. That is almost certainly not the case, but it is an interesting case of convergence of structure. So here's an example of spirochete. This is Treponema denticola. This lives in your mouth. Not a particularly nice one. Treponema denticola isn't like pallidum that causes syphilis, but it is associated with gingivitis. It turns out that it's not a pathogen in the traditional sense, but it is. There's a notion these days that not all diseases are caused by single specific pathogens, but by bad microbial communities. And this is a case of that, where tooth decay or particularly, I guess, gingivitis. The microbiomes of the gingiva can either be positive or negative. And in the cases of negative, then Treponema and some other organisms tend to be much more abundant in these bad gingival microbial populations. And so seeing a bunch of Treponema in your dental plaque is usually a bad sign. Here's Borrelia. Borrelia recurrensis is one of these non-helical spirochetes. So in an EM, you don't see 3 dimensions. You only see basically a shadow. But this organism really is a flat sine wave. Borrelia burgdorfii is the most well-known of these. This is the primary cause of Lyme disease. And a bunch of other ones. Borrelia recurrensis causes a similar relapsing fever, but it's carried by lice rather than by ticks. Really, notice the shape of the cell. Not only is it a flat sine wave, but the tips are tapered like that. That's kind of the diagnostic feature. An interesting aspect of the molecular biology of spirochetes is that linear genomes are the norm, not circular genomes. Circular genomes do occur. Circular plasmids do occur. But generally speaking, both the plasmids and the genomes are linear in spirochetes rather than circular. It's quite common, interestingly, for different strains of the same species to have the same plasmid in either linear form in some strains and circular form in others. The leptospiras are really interesting. Leptospirosis, of course, is an awful disease. Not too easy to identify. The interesting thing about leptospira, this is biflexa, is that their structure is specifically designed to provide motility through really tight, hard-to-get-through environments, such as the interstitial spaces of an animal's body. You know, it's not like you can just swim around in a human body like you're in a swimming pool. You've got to push your way through between cells and in highly viscous, tight environments. So how does leptospira do that? It's a helix, as you can see, and you can see the axial fiber very nicely in this. But it also contains a superhelix at one end. And so this is a large, if this is a corkscrew, imagine you've taken a corkscrew and twisted it around a cylindrical object. And so this is a broad open helix here, in addition to the helix of the organism, and the tail has a hook. And so rotation of the primary helix, and assuming that this hook then is unable to rotate because of the viscous environment, means that this open end here corkscrews its way through tough environments. And in mutants of leptospira that lack that structure, they're still motile in low viscosity environments, but they're not motile in tough ones. Yeah. That's a really interesting question. If you look at that, so the question is, which came first, linear or circular chromosomes? In bacteria generally, circular chromosomes and plasmids are the norm. And if you look at a phylogenetic tree of the bacteria and plant on there linear versus circular chromosomes, it's clear that linear chromosomes are derived. Linear chromosomes also occur in actinobacteria. Linear plasmids occur kind of scattered around the tree. Spirochetes generally have linear chromosomes, but clearly the ancestors, the induction would be that those would have been circular chromosomes. In archaea generally, the chromosomes are circular. Interestingly, they don't have a single origin, they usually have multiple origins. And in eukaryotes, of course, linear chromosomes are the norm, although not think of them as exclusively, that's not true. And so that's a little bit unclear, but probably you would think then that because of the way that archaea and eukarya are related to each other, probably circular chromosomes would have been the norm in the last common ancestor. Cool question, I like that. All right, so let's